Hello children, today I am going to take the topic genetic disorders. They are broadly classified into Mendelian disorders and chromosomal disorders. Mendelian disorders and chromosomal disorders, the first difference is how are they caused. Mendelian disorders are caused due to mutation in a single gene, whereas chromosomal disorders are caused due to failure of segregation of chromosomes at the time of gamete formation, resulting in trisomy or monosomy. Trisomy means 2n plus 1. Monosomy means 2n minus 1. So, let us analyze these two conditions, trisomy and monosomy. So, to analyze that, let us see the normal condition. Normally, take the genetic constitution of a female. She has 44 autosomes and 2 sex chromosomes which are X and X. Males have 44 autosomes with 2 chromosomes, 1 X and 1 Y. So, this is the genetic constitution of a female and this is the genetic constitution of a male. Now, we have learnt according to Mendel's second law, at the time of gamete formation, the alleles separate. So, here we should remember the chromosomes should separate in such a way that the gametes receive only one of the chromosomes, never both. So, now I am going to, the gametes are formed in females, which are the gamete, the egg. So, I am going to represent it by circle. Okay. Now, 44 autosomes are going to segregate. So, how it will separate? They separate from each, they were in a pair. So, they separate such that 22 goes to one egg and the other 22 goes to the other egg. Now, the segregation win of the sex chromosome. One X comes here and the other X comes here. Similarly, in males under normal condition, the gametes produced are the sperms. Sperms are representing symbolically. Now, the autosomes separate, 22 autosomes here and 22. So, of the 44, 22 goes to one sperm, 22 goes segregated to the other sperm. Now, the sex chromosome also gets segregated. In one category of sperms, they take X, the other Y. So, it got segregated, it occurred normally. There is no abnormality. Now, what happens? We have learnt in 10th standard. Now, what happens? If this sperm that is bearing X chromosome as sex chromosome fuses with the egg, what do we get? 22 plus 22, 44 autosomes. And look at the sex chromosome XX. What is this person? This person is a female. You know, XX means female. Now, what if this sperm, that is sperm bearing Y chromosome, fuses with this egg? So, 22 plus 22, 44 autosomes and the sex chromosome XY. What is this? This is a male. So, from the sex chromosome, we can determine whether the child is a male or female. Okay. So, this is a male XY and female XX. So, they have a pair of sex chromosome. In females, there are 2 X chromosome, males as X and Y, and the autosomes 44. So, this condition is diploidy, normal condition. So, here it is represent, normal condition is represent 2n. 2n means 2 into 23, which is equal to 46. This is normal. But now we are talking about the Mendelian disorders where we see, the chromosomal disorders where we see it is deviating from 46. Either it is 2n plus 1. The meaning of 2n plus 1 is 2 into 23, that is 46 plus 1. So, what is it? It is 47. Or 2n minus 1, that is 2 into 23 minus 1, that is 45. This condition where it is 47 is called trisomy, and the condition where it is deviating from 46 and it is 45, it is called monosomy. Why it is called trisomy and monosomy? Trisomy means one chromosome is in 3 copies. Monosomy means one chromosome is in 1 copy. Whereas in the normal individual, see, this is the case of normal, all the chromosomes are in 2 copies. First chromosome, second chromosome, third chromosome till 22nd till 23rd chromosome all are in 2 copies. That is normal. but these are the abnormal conditions. Okay, 2n plus 1 trisomy and 2n minus 1 monosomy are the abnormal conditions, and these are the two categories coming under chromosomal disorders. So, first point I have highlighted, that is the cause of the two sets of disorders. Mendelian disorders is due to mutation. We will be learning mutation in another video, and chromosomal disorders caused due to the non-segregation or failure of separation of the chromosomes at the time of gamete formation or cell division, resulting in trisomy or monosomy. Now, the second difference, pattern of inheritance. The pattern of inheritance the Mendelian disorders follow is the Mendelian pattern. That means it follows the first law of inheritance, second law of inheritance and third law of inheritance. So, it follows Mendelian laws of inheritance. So, the pattern of inheritance it follows, and hence it gets the name Mendelian disorders. But chromosomal disorders does not follow the laws of inheritance. Third point, how we can study Mendelian disorders? We can study with the help of a pedigree chart or pedigree analysis. By pedigree analysis, we can study the inheritance of Mendelian disorders, whereas chromosomal disorders can be studied with the help of karyotyping. Now, let us see what is this pedigree chart. I am zooming through a pedigree chart. See, you can see squares and circles. Okay. So, this pedigree chart has symbols. Okay. The symbols are, you can see square and circles. Square indicates males and circle indicates females. If the square is shaded, usually shaded red in color, then it is affected. But if it is not shaded red in color, means it is unaffected. So, we have males. So, we have two types of males. See, the males which is not shaded means unaffected male, and the male which is shaded red means it is affected male. Now, female represented by circle, unshaded means unaffected and shaded red means affected. Okay. Then, in some pedigree chart we have another symbol, a rhomboid shape. What does it indicate? It indicates that the sex is unspecified. Unspecified sex. That is, when we are tracing the family tree, sometimes the individuals of one generation will not remember whether it is a male or female of that generation, 3-4 generations back having that trait. In that case, we represent it as sex unspecified, which is indicated by a rhomboid structure. Now, again the rhomboid structure, if it is shaded means it is affected. If it is unshaded means it is not affected. Okay. So, this is affected and this is unaffected. So, you understood the symbols used in pedigree chart. Then, look at the pedigree chart. If I am highlighting it with yellow in color, see this dash between the square and the circle indicates mating or marriage between a male and female. But if there is two lines between the two symbols, two lines means mating between close relatives. Okay. Then from this it is very clear they are descendants. Say, take the descendants of these, how many children do they have? They have one boy, again another boy, then one girl and again one girl. Of these, so the first child is this is the second child, third child, fourth child and fifth child. Of these 5 children, all the children are the first, third and fifth child are affected. So, that is why it is shaded red, and the second and fourth child are unaffected. That is why it is blue in color. So, I hope you have understood these are the descendants of the generation. Then again it shows marriage, this indicates marriage and they are descendants. So, clear? So, pedigree analysis is a strong tool used by genetic counselors for tracking the inheritance of various traits, abnormalities or diseases. Okay. So, it is a strong tool used by genetic counselors. Okay. And there are symbols used in pedigree analysis, namely 3 symbols: squares to indicate males, circles to indicate females and rhomboid to indicate unspecified sex. If they are shaded, it indicates the trait in question or they are affected. Unshaded means unaffected. Clear? Then one line between the square and circle indicates mating or marriage between 2 different individuals, but when there are 2 lines it indicates mating between closely related individuals. Clear? Now this side I showed you how to study with the help of karyotyping. So if I enlarge this karyotype, see, so you can see the karyotype consists of all the 46 chromosomes, and analyze each chromosome. See, the first chromosome in pair, second chromosome is in pair, third chromosome is in pair, fourth chromosome is in pair, fifth chromosome is also in pair, sixth, seventh, all are in pairs, which means 2 into 23 means 46. Okay, so they occur. So this is the karyotype of a normal individual. Okay, so this helps to study chromosomal disorders. That's why we take the karyotype of the chromosomes. Clear? So once again, a recap of the difference between Mendelian disorders and chromosomal disorders coming under Mendelian disorders. Clear? Genetic disorders are classified into two types, Mendelian disorders and chromosomal disorders. The cause of Mendelian disorders, mutation in a single gene, whereas cause of chromosomal disorders due to the failure of segregation of chromosomes during gamete formation or during cell division, resulting in trisomy or monosomy. Trisomy means 2n plus 1, monosomy means 2n minus 1, meaning that one chromosome is in 3 copies, that is trisomy. Monosomy means one chromosome is in a single copy. Then pattern of inheritance. Mendelian disorders follows the Mendelian laws, so it follows the Mendelian pattern of inheritance, whereas chromosomal disorders does not follow the Mendelian pattern of inheritance. Then Mendelian disorders are studied by pedigree analysis, whereas chromosomal disorders are studied by karyotyping. I hope you have understood the differences between the two categories of genetic disorders, Mendelian and chromosomal disorders. Now let's go into the different Mendelian disorders. The first Mendelian disorder we are going to study is Down syndrome. And when we are studying, we are going to emphasize 3 points, and the 3 points are starting with alphabet C. So it's easy for you to remember. Genetic constitution, then the cause and the characteristics. Clear? So the genetic constitution of Down syndrome, remember, it is a trisomy. The genetic constitution is 45XX or 45XY. So what is the total? It is equal to 47. So definitely it is trisomy. I'll show you how it has been formed. Second, what is the cause of Down syndrome? Due to the non-separation of 21st autosome. See, we know there are 22 pairs of autosome. Here the 21st pair of autosome did not separate. Now look at the characteristics. Look at this female. You can see the characteristics of this female. I'll just zoom through this picture. See, the characteristic of this female. Characteristics: short statured. Now the head is round and small, partially open mouth, and it's always open but not widely open, partially open mouth and with lots of salivation, furrowed tongue. Then you can see the palm which is very broad. Unlike our palm, the palm is very broad with characteristic palm creases. And worst of all, they are mentally, physically and psychomotorly retarded. So they show all retardation: mental retardation, physical retardation and psychomotor retardation. So once again, repeating the characteristics: they are short statured, the head is round and small, then the mouth is partially opened, tongue is furrowed, the palm is broad with characteristic palm crease, and they show mental, physical and psychomotor retardation. Clear? The characteristics. So genetic constitution, remember, it is a trisomy. That is, the 21st pair of autosome is in 3 copies. Now we can just see, we'll analyze what has happened here. It usually happens in mothers who conceive very late, after 40 years. So this is the genetic constitution of the mother, 44 autosome XX, and she marries an individual male, 44 autosome XY. Now the drawback comes in the gamete formation of the mother. See, eggs are formed. Look, the 44 chromosome should separate 22, 22. But what happens is the 21st pair does not separate. So in one it becomes 23, in the other one it becomes 21. Instead of 22, 22 it becomes 23, 21. And here normal gamete formation, 22, 22 autosomes separated, and one X chromosome here and one Y got distributed. Here definitely only X gets segregated. Now if this sperm fuses with this egg, see what we get, 23 plus 22, 45 autosome XX. See, so we got a Down female. So what female we got? A Down syndrome female. See why she is down? Because the number of autosome has increased, not 44, it has become 45, because the 21st pair is in 3 copies. It should be in just 2 copies, but is in 3 copies. Now if suppose this sperm, the Y sperm fuses with this abnormal egg. So the 2 eggs form. This is an abnormal egg. I am indicating with red to show the abnormality. This is an abnormal egg. Now suppose this abnormal egg fuses with this normal sperm. Again you can see the chromosome number 45 autosome, and look, XY. So this is a Down male. So we saw how the Down male and female is formed. This is a Down female and this is a Down male. Clear? So it happens usually in women who conceive at a very later stage of their life, 45 plus, etc. What happened? The defective gametes are produced, that is eggs, the 21st pair does not separate. So therefore one egg will receive additional autosome 23X. The other also is an abnormal egg, 21X. That's another syndrome which is not needed to study now. Then in the male, the father, the normal gamete formation 22X and the other set of sperms 22Y. If this sperm, X sperm fuses with this abnormal egg, see the genetic constitution, the genetic constitution will be 45XX. That is a Down female. But if the Y sperm fuses with this abnormal egg, see the chromosome constitution 45XY. Okay, so the drawback is with respect to the autosome. Which autosome? The 21st autosome. Clear? I hope Down syndrome is clear. Next syndrome is the Turner syndrome. So the second syndrome is the Turner syndrome. Okay, so first let's analyze the genetic constitution of Turner syndrome. Genetic constitution is 44 autosomes. So the number of autosome is normal, 44 autosome X0. So the problem is with the sex chromosome. Now the cause is non-separation or segregation of sex chromosome. Now look at the features. The last parameter is the characteristics. Look, she is a female. See, she is overall a female but showing masculine characters. That is, the ovaries are underdeveloped, she is sterile and also underdeveloped breast, etc. So she is a female showing masculine features. Clear? Now the next one, the last disorder is the Klinefelter syndrome. Klinefelter syndrome. Now the genetic constitution of this is 44 autosomes. So which means there is no problem with the autosome. The autosome got separated correctly. But look at the sex chromosome XXY. See, so now it becomes total to 47. So it is an example for trisomy. So the second trisomy we are learning is Klinefelter syndrome, where it is deviating from 46, 42 to n plus 1, which is equal to 47. Now the cause is again same reason like Turner syndrome, non-segregation of the sex chromosome. Okay. Now the characteristics, the last C, the characteristics. See, he is a male. So male showing female characters, mainly the female characters enlarged breast and sparse body hairs, like females very less body hairs, not showing so much body hairs like a masculine structure. Okay, such a condition is known as gynecomastia. Okay, males showing feminine characters. Okay, so this, and also they show only slight retardation, not chronic retardation like Down syndrome, mental retardation, slight retardation, but these also are sterile. So the Turner syndrome and Klinefelter syndrome is both the individuals are sterile, they cannot reproduce because of their rudimentary sex organs, and they show the opposite genders characteristic. Klinefelter syndrome, they show feminine characters like developed breasts and sparse body has. Whereas Turner syndrome, a female showing masculine characters. Okay, and both cases they are not showing chronic mental retardation, but they are sterile. Okay, now I will show you how the Turner's and Klinefelter syndrome are formed. How I showed you Down syndrome, I am going to show you in one cross how the two types of syndromes are caused. Okay, now this is a female 44 autosome XX is crossed with the male 44 autosome XY. Again, the problem usually occurs in the case of gamete formation in females. Okay, now you see the gametes that are formed. I am showing it in circles to represent egg, and here I am showing it in symbolic sperms. Okay. Here the problem is not Turner's and Klinefelter syndrome is not due to the segregation of the autosome. The autosomes got correctly segregated. So 44 splits into 22, 22. So here 22, 22. So no, it's not the problem with the autosome, but it's a problem with the sex chromosomes. Now look at the sex chromosome. The two X's came together here. They did not separate. The X chromosome should have separated. One X should have gone to one egg, the other X should have gone to the other egg. No. What happened? Both the X's joined and went together to one egg. So this is an abnormal egg, and this egg is also abnormal, 0. It does not represent O, it represents 0. So two abnormal eggs are produced. This is an abnormal egg. This is also an abnormal egg. But here normal segregation takes place. The 44 separates 22, 22. Then X comes here and Y comes here. Ready? Now, if this sperm, I am highlighting with yellow so you will understand, if this X sperm fuses with this abnormal egg, what egg? 22 autosome, no sex chromosome. Now look at the genetic constitution. We get 44 autosome. The number of autosome is unaltered, normal autosome number, but look at the sex chromosome X0. So what did we get? We got a Turner's female. Turner syndrome, and all Turner syndromes are females. Then see the second case. If this sperm, normal sperm, fuses with this abnormal egg, see the normal sperm 22Y fuses with this abnormal egg. See, look at the abnormality of this egg. Instead of having only one X chromosome, having two X chromosome fuses. Now just see that what will happen when these two join? 44 autosomes. So the number of autosomes is the same or normal, but look at the number of sex chromosome X, X, Y. See, this is a Klinefelter's male or Klinefelter syndrome. And it's always a male. Clear? So I hope you have understood Turner's syndrome and Klinefelter syndrome. See, look at Turner's syndrome. Look at the genetic constitution, and Klinefelter syndrome. Look at the genetic constitution. It all deviated from the normal genetic constitution of 46. See, look at this. It is 44 plus X. So it is 45. So what is this? This is monosomy. This is an example of monosomy. Now look at the second case. It is 44 plus, see, how many sex chromosomes? There should be only 2 sex chromosomes. There are 3 sex chromosomes. So it is equal to 47. So what is it? Trisomy. So trisomy because the sex chromosomes are in 3 copies. Monosomy because the sex chromosome is only in 1 copy. So I hope that you are clear with these these chromosomal disorders coming under genetic disorders. So what I did towards the end, I gave a comparison between Turner's and Klinefelter syndrome. Usually Turner's and Klinefelter syndrome are asked in comparison because it is not an autosomal disorder, it is a sex chromosome disorder. Here the genetic constitution is 44x0 here. The genetic constitution is 44xy giving 47. There it is giving 45. So that is an example of monosomy. This is an example of trisomy. So usually these two are asked as comparison. So these are females, these are males. These are females showing masculine characters. These are males showing feminine characters. In both the cases, what is similar? Both are sterile. They cannot reproduce. Okay, and they only show very slight mental retardation, and both the same reason. The reason is the same, or the cause is the same, non-segregation of the sex chromosomes in both the cases. And this is an example of monosomy, 45, whereas this is an example of trisomy, 47. Clear? I hope you have understood the three syndromes coming under chromosomal disorders. First, Down syndrome, it can be a male or female due to the, here the drawback is in the non-segregation of the autosome. And then we are having the Turner syndrome and the Klinefelter syndrome due to the non-segregation of the sex chromosome at the time of gamete formation. Thank you.